Alrighty, we can go ahead and get started. I've uploaded the slides again right now. Can I just make sure you can hear my screen? Well, hear me and see my screen, not hear my screen. Awesome. Cool. Cool. 32 so far. So as usual, there's probably gonna be some late comers. That is fine. You can hear my screen. I am glad. This is quite the skill. Alrighty. Today we have reproduction and development. This itself isn't incredibly high yield, at least from my experiences. It's more of it kind of pops in under the guise of endocrinology, and so this is it'll be a nice little intro into hormonal pathways for next week when we talk about endocrinology. So I'm going to kind of try to hammer that in. The biggest thing about reproduction and development is just the hormonal control. And so we haven't been too I'm concerned with hormones up to this point, but after today, hopefully, you guys, we're going to get a few hormones under our belt, kind of what they do, where they pop up, kind of the pathway they take. And so the pathway that these guys will take will be very, very similar to a lot of the pathways that other hormones take next week. And so will be a nice little intro to that. Um, Something about repro and dev, if you do get a passage on it, usually what I've seen is there are a lot of questions themselves that could be standalones that are grouped in with a passage, and you don't necessarily need to read the passage to understand it, especially for repro and dev. Repro and dev, a lot of standalone questions like, hey, here's this piece of information. Do you know it? More so than application. You don't need to know too much about development embryology. Thank gosh, that is not something that is fun to learn unless you're into that. Um, but yeah, so we'll go over what the MCAT says we need to know as usual. We'll try to stick with what we need to know rather than focus on a lot of the extraneous information. There's one thing that is in that you need to know that I don't think is in the book. I skimmed the book yesterday, and it doesn't talk much about the placenta and fetal circulation. So we'll talk about fetal circulation, its blood flow, and how it's different from ours. So that's something that is not in the book, but we'll cover today. So let's see how many people are here. Okay, so 3 more people showed up. Let's send the slides and then get cracking. Okay, so slides are uploading. What we need to know. So we're going to go over the different structures and their functions. The gonads, the genitalia, we're going to look at their differences, and especially in the hormonal control. Hormonal control is similar but different. And so the females, as we know, have the reproductive cycle that is on about a month basis, about 28 days long. So we'll go over how hormones fluctuate during that. And we're talking about pregnancy, parturition, which is the fancy term for birth, and then lactation. And then we're going to briefly go over gametogenesis by meiosis. How do we produce our sex cells? And those sex cells are the ovum and the sperm, so the differences in their formation, their morphology or their structure, and what they contribute to the next generation. And we don't need to know too much into development, but we need to be familiar with the stages of early development as we go through cleavage, and then we kind of create smaller cells, which make more sense. They'll move around, create different layers during gastrulation. And then once they do that, we have different germ layers that start to arise into different structures, different tissues. So the structures and the functions. So the male gonads are also known as the testes. And so gonads are really important in creating our sex cells and also in the hormonal production. And so in the male gonads, the male testes, this is where sperm is made, and specifically in the seminiferous tubules. So these little structures, they're not actually yellow in real life. This is just color coded. These seminiferous tubules are the site of sperm production. The gonads also make testosterone, and for the males, they are external organs. The female gonads are the ovaries, and so they house immature eggs until puberty. Right here, the ovaries, there are two of them, they're bilateral. And then each month, one egg matures and will get released into the fallopian tubes. So these are the fallopian tubes, and we'll go over that. The ovaries make estrogen, and they are an internal organ. Let's see. Okay, so other structures, other male structures, other than the testes, include the penis and various ducts and glands. And we'll look at different ducts and glands on the next slide. But as I said, sperm is made in the seminiferous tubules, so these yellow structures right here. And after they get produced, they get stored in this green structure called the epididymis. When they're ready to get released, they leave the epididymis and travel up the vas deferens. And so this little blue tube called the vas deferens is the highway that sperm takes as it leaves the body. And so you actually need to know the pathway at which sperm takes as it leaves the body. It gets produced and then leaves. And so I have a mnemonic for that. That mnemonic is 7-up. So when I'm stressed, right, and I can't think straight, which is pretty much throughout the entire MCAT, right? Mnemonics help me keep things a little bit more straightforward. So everything starts off at the seminiferous tubules, right? Sperm gets made in the seminiferous tubules. So that's S. E, it gets stored in the epididymis. After it gets stored in the epididymis, it leaves out through the vas deferens. That's V. And then after the vas deferens, it enters into the ejaculatory duct. It's another E. N is nothing. And then out the urethra, out the head of the penis. And so the pathway it actually takes, if we kind of zoom out, we have the testes here, right? And we have the epididymis, leaves out the vas deferens, V, goes around the bladder, go through the ejaculatory duct, which is right here, and then out the urethra and the head of the penis. So 7-up for the pathway that sperm takes. Note there are 3 glands that you don't really need to know exactly what they produce, but know that they do exist. There's the seminal vesicle, which exists right here. There's this little guy right here called the bulbourethral gland. It's spelled right there, bulbourethral. Also called the Cowper's gland, and then we have the prostate. And these guys will start injecting fluids along with the sperm, help with lubrication, add an alkalinity to it, and we'll kind of circle around why there's an alkalinity to semen and some nourishment. And so when you do have semen overall, and we'll circle back around to that, it's not just sperm, it's filled with fluids from various ducts and glands. So those ducts and glands are the seminal vesicle, spot right there, the bulbourethral gland, and then the prostate gland. Other than the ovaries, the female structures include the fallopian tubes, uterus, and vagina. So you have, it's bilateral, right? So we have 2 ovaries. You have the fallopian tubes that attach to the ovaries via the vertebrae. We have the uterus, then out the vagina. So that's the path that an egg would take if it wasn't fertilized. We'll talk about fertilization versus non-fertilization today as well. So each monthly cycle, primary oocyte, we'll talk about what a primary oocyte is, for an immature egg will mature into a secondary oocyte. And the secondary oocyte is what gets fertilized inside the fallopian tubes. The lining of the uterus is called endometrium. And so each month, the endometrium will thicken, prepping for a fertilized egg to implant. And if it does not implant, menses will occur and it will shed that lining. So hormonal control, I think it's the next 3 slides. Next 5 slides are probably the most important of today. So hormonal control, most important part of reproduction and development. And so this will be the intro to next week. So next week, we're going to apply the same pathway to several different hormones. So we're going to start off with male hormonal control. And so I'm going to introduce something that kind of helps keep it straight. For all hormones, for most hormones, we follow this thing called, I'm going to upload the slides one last time. We follow this thing called the HPO axis. You might also see it written HPA axis. What is that? So let's see, we have blue right here. So we have the H, hypothalamus, right? Then we have the pituitary. And then we have O, so a target organ. So the hypothalamus will release a specific type of hormone, and depending on what pathway it is, it will release a different type of hormone. But for the reproductive control, that hormonal pathway, the hypothalamus will release a hormone called GnRH, or gonadotropin releasing hormone. So that'll get released from the hypothalamus, which will signal to another endocrine organ called the pituitary. So there's two parts of the pituitary. We're not going to worry about the posterior pituitary today. We'll worry about that more so next week. But the hypothalamus for this axis will release GnRH, or gonadotropin releasing hormone, to the anterior pituitary gland. So H, hypothalamus, P, pituitary. And then the pituitary is going to release hormones to a target organ. And so for this pathway, the anterior pituitary will release gonadotropins. Those gonadotropins are LH and FSH. So LH is luteinizing hormone, and FSH is follicle stimulating hormone. And so good thing about the hypothalamic hormones is they typically do what they're named. So gonadotropin releasing hormone, what does that tell the pituitary to do? Release gonadotropins. Very straightforward, named like that. Question, what does O stand for? It stands for organ. Yeah, so the target organ. So hypothalamus, pituitary, target organ. And so some sources you might see it written as HPA, which what is A? Is the adrenals. And so some might just generalize it and just say HPA, hypothalamus, pituitary, adrenals, but I prefer the O because the O is interchangeable. So in this case, for the testes, it could be HPT, hypothalamus, pituitary, testes. For females, it could be hypothalamus, pituitary, ovaries. So that last one is interchangeable, and we'll kind of change it up and switch it up next week because a lot of different hormones follow this pathway. But for today, hypothalamus releases that GnRH onto the anterior pituitary. Anterior pituitary releases LH and FSH. So those two hormones are going to flow throughout the blood, and eventually, as the blood reaches into the male gonads, the luteinizing hormone will tell the testes to start producing testosterone, and the follicle stimulating hormone will tell the testes to start increasing spermatogenesis. So LH specifically signals on a type of cell called Leydig cells, and FSH on a specific type of cells called Sertoli cells. That may pop up in the passage, the specifics on what cell type it signals to. It's not something I have seen. It could be something that's given in the passage. If I personally, when I studied, I didn't memorize, I memorized it, but I wasn't too focused on what LH signals on and what FSH signals on. I was more of, let's back up and let's look what they actually do, what the hormone actually does. So why would we want to know that? Because they can add an antagonist. They can add in something that blocks LH. So in this pathway, if you have something that blocks LH, but doesn't block FSH, what would you see? And so just knowing that LH stimulates testosterone production. So if you don't have testosterone production, you'll have less of that, but you could still have some spermatogenesis because FSH is signaling. There is the male hormonal control pathway is self-regulating. And what I mean by that is the negative feedback loop, the very, very downstream hormones that get produced, testosterone and inhibin, when they flow throughout the bloodstream and they get, when that bloodstream passes through the hypothalamus, when it passes by the pituitary, it has a negative feedback loop on it. It slows it down. So what I mean by that is if we have too much testosterone, if this pathway is too active and we have too much testosterone, it'll go back and tell it to slow down. So the negative feedback, if you have too much, tells it to slow down. Same with inhibin, which gets produced by Sertoli cells. If you have too much, it'll go back on the pituitary, go back on the hypothalamus, tell it to slow down. And so if it tells it to slow down, we'll start having less testosterone, less inhibin. If you have less testosterone, less inhibin, then you'd have less negative feedback and it can pick back up again. And so it keeps itself in a nice happy medium range because of the negative feedback. And so all negative feedback systems work in this manner. It kind of self-regulates itself. It doesn't get too out of hand, because if it gets too out of hand, it will have negative feedback. But if it's going too slow, you won't have that negative feedback and it will pick back up. What are examples of secondary sex characteristics? So for males, right, chest hair, armpit hair, deeper voice, pubic hair, penis grows, that type of stuff. That makes sense. Yeah, so if you have testosterone, more testosterone will lead to that. This is kind of a tangent, but male pattern baldness, which is it's later on a secondary sex characteristic, right? Because it's male pattern baldness. That's caused by too much testosterone in that area of the hair. So too much testosterone, you'll have a breakdown. I think it's 5-hydroxytestosterone, and that shuts down the stem cell niche of your hair. So testosterone actually causes male pattern baldness. Very interesting. But yeah, negative feedback loop, it regulates itself. Too little testosterone can cause high voice and stuff. It can, yeah. A lot of times there's like enough testosterone for voice to be okay. A lot of times when guys have low testosterone, which is a thing, and you would see an endocrinologist for that. A lot of lethargy, weakness, mood changes, that type of stuff. And so if during puberty there's not a lot of testosterone, then yeah, you'd still have the high voice. You wouldn't really have a lot of the secondary sex characteristics build up at that young age. But usually issues of low testosterone happen in like the mid to late 40s, early 50s. So yeah, so you can take testosterone replacement. So speaking of puberty doesn't happen until you get an LH surge. So the hypothalamus will start releasing more GnRH, whatever age that might be for males, can often average between ages 12 to 17. More GnRH builds up, you start building up more and more LH, and you get an LH surge. Puberty is kicked off by a surge of luteinizing hormones specifically. So the LH surge is something I'm going to repeat quite a bit today because it is a very important thing. The body is really sensitive to LH levels, so LH surges can cause a lot to happen. In males, mostly just puberty, because we do have that negative feedback loop. The LH surge is a lot more versatile in females, and we'll go over that. All right. So this is male hormonal control. I'm going to do something real quick, and let's go over. I'm going to block off half of this. Okay, so can I fill this box with a color? Let's see, or maybe not, whatever. Okay, so for the most part, female hormonal control is the same. For most of the month, it exists in this negative feedback state. And right, again, it's the HPO axis, hypothalamus, pituitary, ovaries. So same hormones, same pathway. Hypothalamus releases GnRH onto the anterior pituitary. The anterior pituitary releases LH and FSH still. And so the LH and FSH signal to the ovaries to start producing estrogen and progesterone. So those are the female hormones. And for most of the month, we exist in a negative feedback state. Too much estrogen progesterone will tell the hypothalamus and the anterior pituitary to slow down. So it keeps it at a nice level. But something interesting happens. Between days of a 28-day female hormonal cycle, around days 12 to 16, on average, it's day 14, it switches. Levels of estrogen and progesterone get too high, and the body switches into a positive feedback loop. And so what a positive feedback loop is, it causes an exponential increase in absolutely everything. So we're still having the GnRH signal to the anterior pituitary. The anterior pituitary releases LH and FSH to the ovary. Ovary releases estrogen and progesterone. But what happens is the estrogen and progesterone, they signal back up on the hypothalamus in the anterior pituitary to speed up. It is an agonist. It tells it to increase its rate. And so it produces more GnRH, more LH, more FSH, which produces more estrogen, which gives it even more of a positive feedback. And so we, around days 12 through 14, we get this exponential increase in all these hormones. And so what this does is this causes a surge in all hormones, but what the body's going to realize, what it's going to recognize, is that surge in LH. If a female isn't at puberty yet, that surge in LH kicks off puberty. But let's say a female's already reached puberty, and let's say they're between ages, 16 and 48, right? What is this going to do? That surge of LH is going to cause ovulation. So ovulation is it's the follicle inside the ovaries is going to release an egg into the fallopian tubes so that it can be fertilized. And so the signal of the LH surge increases fertility because it's releasing the egg into the fallopian tubes. And so after it releases the egg into the fallopian tubes, after days 12 through 14, it's going to go back into the negative feedback loop. There's a question. Prepare for fertilization, hormones increase. Yes, we're going to go over the hormones throughout the cycle on the next slide, and hopefully it makes more sense. But what the hormones are doing throughout the first 12 days is it's maturing the follicle, it's maturing the egg, and then it's going to increase, it's going to go to positive feedback loop, it's going to signal to the ovaries and to the follicle to release that egg, and that signal is going to induce ovulation. Then afterwards, we're going to go back to that negative feedback loop. So we'll look at what picks up afterwards. So yeah, the LH surge induces puberty, right? But then also every month, the LH surge in females is going to induce ovulation. There's a question. The switch to from negative to positive feedback loop is unique to the female reproductive system. Yes, for all 28 days, males will exist in the negative feedback loop, generally quite stable hormonally in that sense. But then females, there's the ovulation where it switches into positive feedback and the hormones increase. And so something that's actually quite interesting is when females ovulate, they're perceived as more they're more fertile, obviously, because the egg will be released, but they're also perceived as more attractive. They did a study where they just had their pheromones were perceived as more attractive. Their voice was perceived as more attractive, ovulation versus non-ovulation. So there's like some built-in mechanism within our body that when female is most fertile, that is when she's most attractive. It's quite interesting. Yeah. So let's actually look a little bit deeper into these hormones. Okay, so we're going to line this up as a timeline, right? So the cycle is on average 28 days. And the reason why I'm saying on average, and when ovulation occurs between days 12 to 16, it's because it's not always regular. It's kind of a finicky system. Some people it's shorter, for some people it's longer. It's not exactly 28 days, but on average it is 28 days. So let's start at day 0. So this entire chart, everything on the left here is day 0. So we're at the start of a brand new menstrual cycle, a brand new start of a 28-day cycle. We are technically in the follicular phase. And so what the follicular phase is inside the ovaries, the follicle which stores the egg is going to start maturing. One of these eggs is going to start maturing. In the uterus itself, we are going to shed the uterine lining from the previous cycle. So it's, there was no implantation occurred because no fertilization occurred. We're going to shed the previous lining. We're going to prep for this new one. In our HPO axis, we're pretty stable on the LH, right? So if we look at this red line, LH is stable. FSH is higher because it's follicle stimulating hormone. It's at this point, it'll be present in higher levels because we are trying to grow this follicle. We're in the follicular phase, right? So we want a follicle stimulating hormone to help stimulate the growth of that follicle that's growing inside the ovaries. And then if we look at the ovarian hormones, so the estrogen and the progesterone, it says estradiol here. So for the purpose of the MCAT, estradiol and estrogen are synonymous. There are 3 estrogens, estradiol, estriol, and esterone, I believe. I think estradiol is also called E3. That is not something that's too important. You don't need to memorize that for the purpose of the MCAT. Estradiol and estrogen are synonymous. It's just a type of estrogen. So those levels are, quite okay. They're quite low and non-fluctuating at the beginning of the cycle. But as we go on, the follicle is going to continue to grow. So as you can see, it's getting a little bit bigger in that ovary. So it's getting bigger, it's getting bigger. We have finished shedding the old menstrual lining. The menstrual flow has stopped. The uterine lining's gone. The follicle is getting big, so we're having a little bit of a decrease in follicle stimulating hormone. We don't need to grow it as fast anymore. But there's something that's quite interesting. The ovaries are going to, now that this follicle is getting big enough, the ovaries are going to start producing more estrogen. And estrogen levels start to get high enough. The hypothalamus and the anterior pituitary are going to switch from that negative feedback to that positive feedback. So that positive feedback is going to cause that exponential increase in everything, right? So you get that exponential increase now in both the FSH and the LH because, right, if you're having that positive feedback and you downstream, the pituitary is going to release more FSH and LH. The body's going to recognize specifically this exponential increase in LH. LH was pretty much stable up until this point. But now we're around, day 12, around day 14, we get that surge, that exponential increase in the LH. Ovaries recognize that, and we're going to release the egg. And so the egg is going to leave the ovary, it's going to enter the fallopian tubes. So at this point, with the surge, the uterine lining is going to start to thicken. It's going to say, hey, an egg just got released. An egg just got released. We need to start prepping for it. We need to create an environment that can house an egg. After we do have that surge, the estradiol levels are going to dip off a little bit again. And as that dips off again, we're going to enter back into negative feedback loop. And these levels are going to dip back down. The FSH and the LH levels are going to dip back down. The egg itself is going to be in the fallopian tubes. It's not going to immediately enter into the uterus. But we're also in this phase now that we have had ovulation. We're in this phase called the luteal phase, because what was the follicle, right? What housed the egg is now going to form this structure called the corpus luteum. So the corpus luteum becomes this temporary endocrine gland. And this corpus luteum is going to release some more estrogens, and it's going to release a hormone called progesterone. And progesterone is that hormone that's telling the endometrium to start to thicken, the uterine to start to thicken. So this corpus luteum is going to start producing more of these hormones. The progesterone and estradiol are going to increase. We're going to thicken the endometrium, waiting for a fertilized egg to implant. But in this case, let's say there's no fertilization, no implantation. What's going to happen? Well, the corpus luteum is going to degenerate, right? It's going to shrivel up and it'll waste away. It'll stop releasing progesterone, estradiol. Because implantation didn't occur, there's going to be no replacement estradiol or progesterone, because we'll see that the progesterone starts to get made after implantation occurs. So if no implantation occurred, the corpus luteum dissolves, goes away. Those levels are going to drop back down because now nothing's producing it. As those levels go back down, that tells the body, hey, no implantation occurred. We need to repeat the cycle. And so as these levels go back down, we see they're back down now at day 0. The body's going to restart. It's going to shed the lining that did exist, that was made in the previous cycle, and another follicle is going to start to grow. We're going to enter the follicular phase of the next egg, and we're going to repeat the cycle on an average of every 28 days, with an LH surge or ovulation occurring around day 14. So around these areas, around this time frame, this is where the female is most fertile, because you do have an egg that is ready in the fallopian tubes that was just released, that is brand new. Any questions on that thus far? This is probably the most important slide of today. Are mood changes that accompany PMS associated primarily with the surge in progesterone? So I think it's the fluctuating of both the progesterone and the estrogens. So I can't attribute the mood changes to just progesterone. I'm not 100% sure on that, so I don't want to give you a wrong answer. I think it's the fluctuation of all the hormones in general that can cause the mood changes. Do I know ish the numbers, like the ranges, like the concentrations? I do not know the blood concentrations. So serum concentrate, you might see it written as blood concentration, serum concentration. So serum is one of those things that you might see on the MCAT. Serum just means blood levels. So does birth control supplant this? Yeah, so there's different types of birth controls. One of them is progesterone. And so what happens is it artificially keeps progesterone levels high. And if progesterone levels don't drop off, then what doesn't happen? I'll let someone give that a shot in the chat. If hormonal birth control keeps progesterone levels high, the absence of the drop off in level, yeah, it'll keep the hormone levels high, and it's not going to tell the body that, hey, no implantation occurred. So we're not going to reset. And if we don't reset, what's going to happen is we're not going to shed the lining. So the lining will stay there. And we won't release another follicle, because it thinks pregnancy happened. So what hormonal birth control does is it kind of tricks the body into thinking it's pregnant, so it's not going to shed the uterine lining. It's not going to release another egg. And because it doesn't release another follicle or an egg, that is what the birth control does. Yeah, so that's a good question. How do you get periods then? The body, you get periods. It also depends on the type of hormonal birth control. Some of them don't use progesterone. And so periods happen at that point. I think it's like every 3 months. So some of them don't use progesterone themselves. Some of them, its progesterone levels aren't as high as they are during pregnancy. And so that might kind of trigger the shedding of the uterine lining, but they don't drop low enough that a follicle gets released. Sometimes some birth controls do like 3 months on and then 1 month off. Yeah. Yeah, no egg is released during those. Let's see. Birth control patch. There is a type of birth control that kind of gets implanted in the arm, and that one releases hormones as well. So it could be patch, it could be pill, it could be an implantation. Yeah, some that are estradiol, maybe that triggers shedding. Yeah, I'm not an expert on birth control. I'm most familiar with the progesterone birth control, and so I know what that one does. I think some of them are estrogen and whatnot, so I'm not exactly sure. So does menopause happen when there are no more eggs? We'll go into that. Yeah, so the menopause happens due to follicular attrition. So we'll talk about the egg buildup and and whatnot a little bit later. But males can regenerate sperm. Females are born with all the follicles they'll ever release, and so as we get fewer and fewer follicles, which is called follicular attrition, um, the ovaries themselves become insensitive to the gonadotropins. So they become insensitive to LH and FSH. And so you start not going through this cycle anymore. So technically, menopause is a time frame of no periods or no, menstrual cycle reset or no ovulation within a year. So it's hard to say that you are in menopause because you don't actually know. A lot of the symptoms of like the hot flashes and whatnot, when people say they're in menopause, it's usually post-menopause. And so it's one of those terms that you don't really know you're in menopause until you're post-menopausal. Why does menopause happen? Yeah. Well, we'll circle back around to this. Don't worry. We're getting ahead of ourselves. So ovulation, right? So it used to be assumed that during ovulation, an egg ruptures and it gets shot out into the fallopian tubes. But that's not exactly what happens. This is a picture on the right. During a hysterectomy, they were removing, I believe this was in Italy. I don't know exactly. I'm trying to recall. They were doing a hysterectomy, removing the uterus, and they caught ovulation occurring. So what happens is we have the ovary, this is the ovary, right? And this is the egg. It kind of like oozes out and goes into the fallopian tubes. This is just an interesting picture of ovulation happening. And so again, it happens between days 12 and 16, the menstrual cycle. So I thought this is a very interesting picture, so I added it in quite a while ago, but I believe this was in Italy, I think like 2 years ago, maybe. But yeah. Okay, let's say pregnancy does happen. Right, so obviously we're not going to want the uterine lining to shed. We're going to want to keep it because if we shed the uterine lining and pregnancy and fertilization and implantation did happen, then that fertilized egg will be shed with the old lining, and we don't want that. So fertilization occurs in the fallopian tubes, and let's say the zygote, which is now the fertilized egg, implants itself in the uterine wall. The outer layer of that egg will grow into the endometrium and produces a hormone called HCG, or human chorionic gonadotropin. So in pregnancy tests, you can either urine pregnancy tests or blood tests. What they detect is HCG. They detect human chorionic gonadotropin. So if we have that in the body, then we know, hey, pregnancy happened. So the HCG is going to be the driver now. It's going to increase estrogen and progesterone levels. And so increasing those levels will keep the uterine lining from shedding. So HCG is now this guy that is taking over and making sure all these levels remain high. During the first trimester of pregnancy, pregnancy is about 40 weeks long. During the first trimester, the placenta forms. We're going to talk about the placenta today. It's quite an interesting organ. So the first trimester, the placenta forms, and you've got some starting of some internal organs. Second trimester, more internal organs develop and continue to grow, and this is when the fetus becomes active. Then the third trimester, you get rapid growth, and the fetus reaches full size. So the human gestational period, or how long pregnancy occurs, about 40 weeks, which is short for us. It shouldn't be that short. It should be much longer. If you realize human babies, when they come out, we're quite helpless. We can't do much. We're quite reliant on parents and those around us. I was watching a nature documentary, right? And it was an elephant giving birth. And this is really weird, but this kind of sparked an interest. And it's like this elephant gives birth, and the baby elephant gets up and walks around and is pretty self-sufficient. It's not 100% self-sufficient, but it could do things that human babies can't do. I was wondering, why is it like that? When we get born, we're just so helpless. And the reason why is because we come out of the oven just a bit too early. The human gestational period should be about 18 months long, or a year and a half. Instead, we're at 9 months. And so the reason why is because as humans started to develop, our heads started getting too big. Our brains are, way too large. And as our head started to get bigger, it started to become dangerous for the mothers, right? Going through the pelvis, it became a lot of damage. You have a lot of mortality during childbirth. And so it was selected for that. The women who could have more babies were the ones that pushed them out sooner. And so evolutionary pressure made us come out kind of half-baked. Typically, if we were in there the entire time, we would come out, more developed. We'd be able to hold up our own head. Our brains will be a little bit more developed, but by the time we get to that point, we're too big to actually come out of our mothers. So that is why our gestational period is so short and why we come out essentially half-baked. So if you're just curious, this isn't something you need to know for the MCAT. This is more for your curiosity. Different stages of birth is when different organs start to develop. So the brain and the spinal cord are amongst the first things that form. And then teeth and the palate are amongst the last. Do you think this trend, hold on, there's some questions. Sorry, there's a lot. Do you think this trend would be involved with animals that consumed larger amounts of calories? I don't know if it's the calories specifically that cause that, because if you think elephants being so large, they need to consume a lot of calories. Again, horses, they function out of birth. They're large, they need to consume a lot of calories for especially the muscle mass that they have. So it's more of the brain size and the head size of trying to push that out through the pelvis. And it's humans just relative to the rest of our body, relative to, human females who their pelvis became smaller, like all, humans, our pelvis became smaller so that we could stand upright. And eventually there had to be a trade-off of, our head became so large and pelvis is smaller because we could stand upright. We had to kind of pump them out half-baked. Yeah. So I guess, evolution pushes to allow for more calories to be allocated towards greater brain development, especially in humans. Humans are quite interesting. If you look at the history of humans and how our brain sizes have changed, especially with the cultivation of wheat. Yeah, that's when our that and fires when our brains started to get big and our gestational period started to shorten. Okay, end of the 40 weeks, we enter parturition. So as I said, parturition is the fancy term for birth. So at the end of gestation, that baby's getting big, right? So the baby's going to add some tension to the uterus. It's getting bigger, and as it pushes on the uterus, pushes on the muscles, the estrogen receptors in the uterine wall will start to bind to a hormone called oxytocin. So oxytocin is a fascinating hormone, and it is so versatile. It does so many things. We'll kind of circle around to oxytocin next week during the endocrine lecture, but oxytocin is very interesting. In this case, it binds to the estrogen receptors in the uterine wall, and it binds because it's getting released due to the stretching of the uterus, and it's going to cause muscles to start to contract. And as muscles start to contract, it signals for the release of more oxytocin. So more oxytocin will bind, cause more contraction. That more contraction will release to the brain, say, hey, let's release more oxytocin, and become a positive feedback loop. So you get more and more and more oxytocin. And that positive feedback loop is going to initiate labor. So the first stage of labor, the cervix of this little tissue barrier where the head comes out is going to thin and dilate. And so it's going to dilate to about 10 centimeters. Once we're at 10 centimeters in dilation, that's where we're going to start pushing and allowing for the baby to start entering the second stage of labor, where contractions become stronger and the mother starts pushing and the baby is expelled from the uterus. And then the third stage of labor, the placenta, amniotic sac, and the remainder of the umbilical cord are going to be expelled from the uterus. And so that's the placenta, which is shown in this bottom right, is very interesting. Some cultures, it's kind of like it's a friend to the baby, right? So we're going to talk in the fetal circulation more what the placenta does. It's the barrier between the mom and the baby. It's where their blood flow kind of acts as that barrier. Some cultures bury the placenta after birth. They keep it, bury it. Typically, in our culture, historically, we just throw it out. That's starting to change. It is an excellent source of stem cells, embryonic stem cells. There's a lot of it in the placenta, so that's being researched and studied on. Some people consume the placenta. So some animals, after they give birth, will eat the placenta. But some cultures here they do as well. But it's starting to become more mainstream to eat it. For example, one of the Kardashians, I don't know which one, um, you know, cooked their placenta and made smoothies out of it. I have a coworker who's pregnant right now, and she is about to give birth. She was talking to me on Thursday saying that she wants to do this thing on TikTok, which I'm not on TikTok, so I can't really comment on it. But apparently, it's common to encapsulate it. So what happens is they cook it and make it into a powder and make pills. And people that she's taking placental pills or wants to. And I guess TikTok, which is advertising that it increases milk production, increases energy, decreases postpartum depression and whatnot. There's not a ton of research on it. I looked into it. So it's something that is one of those things a little bit questionable. If you want to do it, go for it. No judgment. Make sure it's done right. It can be very dangerous. I did find a paper on where if you have bacteria in it that doesn't get properly prepared and you eat it, the bacteria can get to the baby through the breast milk and cause a lot of harm. So just in case you want to do that, just a public service announcement, make sure it's done right. Some people is starting to become more popular here in the United States. It was Courtney. Okay. For some reason, I thought it was Chloe. But okay. Bacteria has found its own niche. It has. Let's see. There's a question. Oh, the Radiolab podcast. Oh, yeah. So have you guys heard of Radiolab? They have an excellent podcast on the placenta. Maybe we can talk about that later. There is a question. Oh, Lord. Your wording of this question is a bit strange. So I'm going to reword this. The female bodily structure of curvy hips are they more preferred often because just evolutionarily, that's why some people, I'm not trying to objectify at all. Some people refer to it as childbearing hips. And so typically, yeah, wider hips, do pique interest more because evolutionarily they would be more successful in preventing death during childbirth. That's not really an issue anymore, but it is within hardwiring that is a little bit more preferred. So that is a thing. It's not necessarily better baked babies, but just you can, you know, potentially have more offspring, and that betters our genetic, propagation throughout. Keeps our genes flowing, and that's what evolution wants. It wants our genes to keep being propagated. Let's see. Yes, we'll kind of go through the placenta a bit more in a bit during fetal circulation. But yeah, after birth, right, we're gonna have lactation. So this is a hallmark of female mammals. Babies don't really need to eat anything. Their all their nutrition is provided in mother's milk. So lactation itself is under endocrine control. During pregnancy, more and more prolactin gets produced. Prolactin, which we'll circle around to next week, is involved in milk production. And so the mammary glands will start producing more and more milk. Get ready. Once the baby is born and starts to suckle, oxytocin will get released. So here we see oxytocin again. Oxytocin is involved in milk letdown, or milk ejection. So prolactin doesn't cause milk to be released. Oxytocin does. And so prolactin is just milk production. Oxytocin is the release of the milk from the mammary glands and into the baby. So the process of which mammary glands begin to producing milk, and it starts during late stages of pregnancy, is called lactogenesis. The very first milk that the baby consumes is called colostrum. So colostrum is very fascinating. It has a higher level amount of white blood cells, antibodies, and what this does, it's heavy in immunoglobulin A. We'll talk about immunoglobulins next week. It gives that baby an immune boost. So the colostrum, not only does it have the nutrients that it needs to help the baby grow, but it also has an immune booster. There's some anecdotal evidence that when a baby is sick, the mother's body can sense that the baby is sick, and it will temporarily give an immune boost. It'll give more immunoglobulins, it'll give more white blood cells to help the baby fight any infection. If a mother gets, there was a study I read last November at this point, where if you did have COVID, the COVID antibodies are detectable in breast milk. And so you could essentially give your baby a little bit of a defense against that. And so it's quite interesting. Let's see. Oh, there's a question. Doesn't the mother's microbiome influence the baby because the baby is placed on the mother's chest right away? So yeah, the baby comes out pretty clean. There's hasn't been exposed to anything, right? It's been relatively safe in the womb. So as it comes out, it's going to have the immune boost from the colostrum. But yeah, the mother's microbiome and her germs will be exposed to the baby, and the baby will have a very similar microbiome. Why is it painful for women when they are unable to breastfeed after pregnancy? Because the buildup of milk gets quite painful, and so that's why they have to have it be released. So that is a problem. So you think prolactin interferes with dopamine in some manner, and as a result, people on antidepressants can experience lactation. I have not heard anything about that. Wow, that's quite interesting. I'll have to look into that. People on antidepressants can experience lactation. That's strange. I've never heard that one. I'm going to look into that one. I'm going to screenshot that real quick. Cool. All righty. Keep on cracking away. gametogenesis. So we're going to back up. We're not even at the baby point yet. The production of our sex cells. And so hopefully you've heard of mitosis and meiosis. Mitosis is right. We have cell division, but the number of chromosomes don't change. We'll be at 2n or 46 for humans, right? But to create our sex cells, we need to enter through meiosis. And so there's two steps of meiosis. And after the first step of meiosis, or meiosis 1, we have our chromosomal number. We go from 2n to n. So our sex cells have 23 chromosomes. And because they have 23 chromosomes, we can take our very downstream sex cells and combine them with another individual's sex cells so that two cells of 23 can combine and create one cell of 46. And that one cell of 46 can continue to divide and can become a full-grown baby. Can I explain independent assortment versus law of segregation? Right now, no, I cannot. I'm just going to be completely honest. I'd have to refresh on that. I may be able to do an office hours where I can go through genetics and explain those if needed, but right now I can't off the top of my head. I apologize. Alrighty. Male spermatogenesis. So the creation of the male sex cells, or male gametogenesis. So because it is the male gametogenesis, we call it spermatogenesis. So this, again, where does sperm production occur? It occurs in the seminiferous tubules. So male spermatogenesis will occur in the seminiferous tubules as well. We start off with the spermatogonium. So the spermatogonium is the germline stem cell. So germline is, what will become our sex cells. So if you ever see germline, those are what become our downstream sex cells. And so our germline stem cell for males is the spermatogonium. Something that's unique about the spermatogonium is that it functions essentially the way most other stem cells do. So what I mean by that is as one spermatogonium enters into mitosis, it's going to create two daughter cells. One of those daughter cells will enter meiosis. And when it enters meiosis, it'll become sex cells downstream. But one of those daughter cells will be able to replace the spermatogonium. It's self-regenerating. So as one spermatogonium splits, one daughter cell will enter meiosis, one daughter cell will replace that original spermatogonium. And so essentially, the male sperm supply is never ending because it can replace its own stem cell. And that's what stem cells do, most of them. Stem cells can replace themselves. And so let's talk about the other daughter cell, where that one that enters into meiosis. So that one, as it finishes meiosis 1, it is now halved in chromosomal number. It's now a 1n. And after meiosis 2, we'll have spermatids that go spermiogenesis, and they grow the characteristic tail that we see in sperm. So this only occurs after puberty. So what is that signal that, in the chat, what is that signal that kicks off puberty? LH surge, exactly, awesome. So the LH surge in males will kick off this process. Do egg stem cells do the same thing? No, they do not, and we'll get through that. So yeah, we'll circle back around to that. Next slide. So sperm is only one component of semen. So people often use them interchangeably. They're not exactly synonymous. Sperm's only one component. It contains fluids and proteins from those various glands that we passed by. So right, those glands, the bulbourethral gland, the seminal vesicle, the prostate gland, right? So some of that fluid is going to add an alkalinity to the semen. So why would we want alkalinity to the semen? Alkalinity is a basic environment. And the reason why is because the female reproductive tract itself is on the more acidic side. So semen will be on the more alkaline side, so that as it gets into the acidic environment of the female reproductive tract, we can increase survivability. If we don't have the alkaline environment, the acidic environment of the female reproductive tract will kill it off. Alrighty, let's go into female oogenesis, but real quick. So does this mean that males experience more frequent LH surges than females? No. So the original LH surge in males occurs at puberty, and it exists in a negative feedback state for just essentially keeps on going. And then this process just never stops. And so it's the initial LH surge kicks this off, but it keeps on going. So we just have that initial LH surge. Females undergo it more frequently because they need it to basically send one of their sex cells into meiosis II. So we'll go back to that. Females, yeah, females have a more frequent LH surge, and that exists on day 14 of the reproductive cycle, and that is called ovulation. Okay, female gametogenesis, called oogenesis. So this occurs in the ovaries and does continue in the fallopian tubes. The female stem cell, the germline stem cell, is the oogonium. So if we look at this, the oogonium, it undergoes mitosis, but one of those daughter cells doesn't go back to replace the oogonium. So what does that do? That means there's a finite supply. If we don't go backwards to replace it, then once we use up all the oogonium, that's it. So females are actually born with all the follicles, all the eggs they'll ever be able to possibly release. So as each monthly cycle passes, the total number of follicles and of eggs will decrease. It's not going to replace itself. And that's where you get the follicular attrition. So for menopause, you lose enough of them, your ovaries become insensitive to the gonadotropins. And so that's what kicks off menopause. But the oogonium is going to undergo mitosis, and it's going to create a bunch of oogonia. Those oogonia are going to grow. So you're going to have some cell growth, and those are going to become primary oocytes. So primary oocytes arrest in prophase 1. So at the very start of meiosis 1, we haven't quite became just N yet. So it arrests at 2N. And so when a female is born, what's inside the ovaries are all these primary oocytes. So at birth, if I get the annotation tool, this is where it's going to arrest. It's going to stay here. So we're going to stop here. All of the primary oocytes will ever get released exist in the ovaries. So what happens is every month, what's going to happen during the follicular phase, one of these guys is going to come out of arrest. It's going to enter into meiosis 1. And then once the LH surge happens, the secondary oocyte ruptures from the follicle and enters into the fallopian tubes. So at birth, the primary oocyte all exists in the ovaries, and then every month, one secondary oocyte one primary oocyte will come out of arrest, enter meiosis 1, become a secondary oocyte during the follicular phase, and that secondary oocyte is what gets released from the ovaries into the fallopian tubes. So this secondary oocyte is going to continue into meiosis 2 in the fallopian tubes. It's going to continue to start, dividing and whatnot, but it's going to arrest at metaphase 2. So it's not going to completely complete meiosis 2, actually. It'll stop somewhere around here. If it gets fertilized, then it'll come out of metaphase 2 of meiosis, and become the final ovum. And the ovum itself, the nuclei of the ovum and the nuclei of the sperm are what fuse to create the zygote. So let's see, there's some questions. Yeah, you only use a super small amount of total you have. Infertility can be associated with hormones indeed. Let's see. Yeah, so you don't use all of them. You don't use all of them up. And at what point do your ovaries become insensitive to, to those gonadotropins? If there's a thing called double ovulation, where you can potentially release two primary oocytes can come out of arrest during the follicular phase, and you can eventually during ovulation release two secondary oocytes. That's called double ovulation. That can happen, and that's how you get fraternal twins. Let's see. Yeah, and so what are these things called polar bodies? So after we enter in meiosis from the females, we create that split and you get these daughter cells. As you can see, you have a secondary oocyte and a polar body. So typically when you get cell division, you have two daughter cells, right? They split everything. What happens is that these polar bodies, don't get any cytoplasm. All the cytoplasm is in the secondary oocyte. So you don't have this cytoplasm super small. It doesn't do much good. What happens, this guy shrivels up and dies via apoptosis. These guys don't really do anything. And so then it's going to split. And as it finishes meiosis 2, you will have the ovum and you have a polar body, right? There's no cytoplasm. There's not much to have. These guys eventually die via apoptosis. So the guys we're interested in are the ones who get all the cytoplasm, and don't apoptose. Does the secondary oocyte get the cytoplasm from the polar body so it is extra big? Yeah, the reason why it's so small is because it's split. This guy stole all the cytoplasm from this guy. So that's where the cytoplasm went. That's why this guy's so big. But yeah. Alrighty. So yeah, whenever that, after this gets released during ovulation, it arrests at metaphase 2, and it will hold here until it gets fertilized. And after it gets fertilized, it'll continue and become the final ovum. No, it doesn't proceed to meiosis 2 only after fertilization. The secondary oocyte, after it gets released, will continue into meiosis 2, where it holds at metaphase 2. So it's already in meiosis 2, but it just pauses in the middle of it, and it doesn't quite finish it up until fertilization happens. So if the fertilization doesn't happen, it won't finish meiosis 2. It's not viable. It can't become a baby. So those final guys that get made are the ovum and the sperm, right? So differences in morphology or structure. I need to clear up the annotation. Cool. So the sperm itself, shown on the right, it has a flagella. Because it has a flagella, it's motile. It could swim around. The flagella also called a tail. It has a lot of mitochondria to power that tail, and that mitochondria exists in the midpiece. It doesn't exist in the head. So why? I'll circle back to that in like 30 seconds. The egg itself is round, non-motile. So the egg here is on the left. So the relative contribution to the next generation. So as the sperm enters the egg, the head will break off, and it's only going to give the nucleus to the egg. So the only thing that the sperm contributes to the next generation is DNA from the nucleus. All of the organelles And everything else, the cytoplasm, it's provided by the egg. Because what's going to happen is the egg itself has all its organelles, it has its cytoplasm. As the head breaks off of the sperm and it goes into the egg, the nuclei will fuse and it will become a zygote. So as we become that 46 chromosome number, as you enter 2N, we start to divide everything that exists inside the cell, meaning the organelles, that comes from the egg. The only thing that sperm does is it contributes DNA. The mitochondria in the midpiece gets destroyed by the egg. So that's if you have mitochondrial DNA, so the mitochondria themselves have DNA, that is only inherited by the mother because the egg is what provides the organelles. So mitochondrial DNA is strictly passed down through the mother. So the sperm itself is motile because it has this flagellum, it has this tail, and help it swim. And it is powered by this mid piece, which is jam-packed with mitochondria. That's right, mitochondria is the powerhouse of the cell, it gives it all its energy. Yeah, so the sperm itself is motile, meaning it can move because it can swim with that tail. All right. So the reproductive sequence. Step 1, fertilization, right? The sperm and the egg meet and they form the zygote. Yep, the first sport we ever played was swimming, indeed. That is true. So fertilization, sperm and the egg meet, create a zygote. That zygote in the fallopian tubes will start to divide. You'll have cell division without cell growth. And what I mean by that is this egg is going to divide, but the overall ball of cells aren't going to get bigger. So it divides, but you're having no cell growth. These individual cells that start to arise because of cell division, they're not going to grow in size. And so that original size of this zygote will be the final size of this dense ball of cell called the morula. So as we have a lot of this cell division, we keep getting more and more cells, 2 turns to 4, 4 turns to 8, 8 turns to 16. You get more and more cells, but the cells themselves aren't getting bigger. So you get this dense ball of cells called the morula. And this all happens during the egg itself, or while that fertilized egg is in the fallopian tubes. While it becomes the morula, it'll start to hollow out. And so that ball of cells is going to start to hollow out and it becomes a hollow ball of cells. That hollow ball of cells is called the blastocyst. And the blastocyst itself is what implants itself into the endometrium. So a zygote doesn't implant itself into the endometrium. The morula doesn't. The blastocyst, when it gets to the blastocyst stage, that's when it implants into the endometrium. And all this cell division occurs in the fallopian tubes as it keeps on going. I just want to say, y'all spitballing. Yeah, so there's a lot of good brainstorming here. I enjoy it. Makes my job interesting. But let's say we become the blastocyst way too soon. Let's say this fertilized ball of cells is moving slowly and you accidentally have this blastocyst that implants itself right here in the fallopian tubes. This happens in about 2% of all births. That's called an ectopic pregnancy. So as it implants itself into the fallopian tubes, right? These fallopian tubes are tiny. They don't have the space nor the flexibility to compensate for a growing ball of cells. And so these are very, very dangerous. As it keeps on getting bigger and bigger and bigger, it can cause rupture to the fallopian tubes. And that is not good, not what we want. About 2% of all pregnancies will be ectopic. And that is not something we want. We wanted to implant in the uterine lining, in the endometrium at the uterus. So that blastocyst is the kind of the first stage of development that we really want to look at here. Is that painful? Like, would you know? I don't know if it's painful. It probably is painful. I cannot comment on that. I actually haven't met anybody who had an ectopic pregnancy. Don't people not know they're pregnant if it's ectopic sometimes and randomly give birth? That is, I don't know about that. I know there's some people who don't know that they're pregnant at all, and they just give birth. I don't know why that happens. I have not looked into the reason behind that. For the MCAT, you just need to know the average person, and people who give birth randomly are not the average person. So that is a good thing you don't need to know that. I don't know that off the top of my head and why what goes on. The baby's not, too terribly big, so they don't realize, oh, hey, there's something growing. But yeah, so in development, that blastocyst is what implants. It's that hollow ball of cells. And what happens is going to start after it implants, it's going to start folding on itself. It's going to folding, and these stem cells will migrate. And you create different layers. Creating different layers is called gastrulation. So gastrulation is going to have these different layers of cells in this giant ball of cells, and these different layers will become different organs. So you get organogenesis. Those different layers will start to arrange and become different organs, and we'll talk about which layers become which later on. Yeah, so pregnancy test will tell you whether you're pregnant or not because you will have that producing HCG. Yeah, so you do have that ectopic pregnancy, it'll burst, cause internal bleeding. I honestly can't tell you if there's ever been a successful baby that was ectopic. The fallopian tubes are very, very small, so it's highly doubted, but I've never looked into that question, so I cannot deny that. I don't want to say with absolute certainty that it hasn't happened. That is interesting. TikTok is it's strange, but yeah. So I couldn't tell you whether or not. So pregnancy tests only detect HCG. Not necessarily. We only look for HCG because it's the best thing to look for. So we pretty much deal with HCG because it's reliable and we can detect it. And so typically, if you go to the hospital, you want to test it, they'll do an HCG test. So that is pretty much the gold standard. That's why we test it. And you could probably look at different progesterone levels and estrogen as well, and know, but you do get fluctuating levels of progesterone due to the corpus luteum. But the HCG is only present during the pregnancy, and so that is why we look for it. So birth. This is what we're going to talk about fetal circulation. So after birth, after we have development and we have given birth, we have to switch our blood circulation. Because when you're in the womb, you're not breathing, right? You're in just this amniotic fluid. You don't need oxygen, right? Your lungs aren't grabbing oxygen from the air. And the lungs are really important in oxygenating deoxygenated blood. And if you recall back to the heart lecture, right, there's two sides of the heart. One side of the heart is oxygen rich, one side is oxygen poor. And the oxygen poor side sends the blood to the lungs to get oxygenated to come back. But because we're not breathing, our lungs aren't doing that in the womb, it really doesn't matter right or left heart. The blood's just all kind of flowing around, all circulating. So we need to switch from this, we're getting all our nutrients from the placenta, to, hey, we're getting our nutrients in the form of oxygen from our own lungs. So there are three structures that exist in fetal circulation that don't exist or shouldn't exist in our circulation now that we're born. The first is the ductus arteriosus. The second is the foramen ovale. And the third is, there it is, ductus venosus. So the first, let's talk about the ductus venosus. So we have an umbilical cord, right, that goes and it connects to the mom, specifically the placenta. The placenta is this organ that it doesn't belong to the mom. It belongs to the baby. So this is the baby's tissues. Genetic wise, it's half mom, half dad. So this itself causes issues because if you have something that is non-self in the body, the mom will attack it. The mom's body would attack it, but during birth, during pregnancy, the immune system of the mom is decreased. It's lowered, so we don't have the autoimmune issue. But this placenta exists as a barrier for the baby. The mother's blood vessels pass through the placenta. The baby's blood vessels pass through the placenta. So they're not connected. The mother's blood doesn't go straight into the baby's blood. But if we use the different concentrations, right? The mother's breathing has oxygen-rich blood. The baby is using up the oxygen, is not breathing, so the blood is relatively oxygen poor. The oxygen from the mother's blood will diffuse across the placental barriers into these chorionic villi and enter into the baby's blood. So the oxygen will diffuse from the mother's blood into the baby's blood. And if you recall back from the cardiovascular lecture, you have this thing called fetal hemoglobin. So the hemoglobin of the baby itself wants to rip off oxygen anyways. So the hemoglobin of the baby, of the fetus, has a higher affinity for oxygen and can pull oxygen off the mom's hemoglobin. So all these different nutrients that are high in the mom's blood, because she's able to eat, because she's breathing, will diffuse into the baby's blood because the baby needs it and it can't get it itself. So the concentrations will be lower. But as the baby's using up these nutrients, he's also creating wastes. And so as it creates wastes, the high concentration of the waste in the placenta from the baby's veins will diffuse into the mother's blood, and the mother's body can get rid of the waste at the level of the placenta. And so what happens is you have this umbilical artery that takes blood to the baby. The umbilical veins help take the blood to the placenta. But where it enters into the body is next to the liver. Next to the liver, there's a structure called the ductus venosus that the umbilical cord takes the blood supply to. So everything that comes from the placenta, comes through the umbilical cord, will drain into the baby's blood supply at the level of the ductus venosus. After birth, we cut the umbilical cord, right? So there's going to be no blood flow through this ductus venosus. The ductus venosus will collapse and shrivel up. So when you guys are in medical school and doing, cadaver extractions, and a lot of people, when you go in and look, you can find someone's ductus venosus. It'll be this artifact that just exists there. It's just this old artifact that's hanging off the blood vessel. It's shriveled up, it's atrophied, so it's not functional, but it's something you can find. At the level of the heart, there's a structure called the foramen ovale. So what is the foramen ovale? So if I draw the heart as a box, that blue is hard to see. Let's look at this red. That red is a little bit better. We have box. Cool. So right here we have the right atrium, the right ventricle, the left atrium, and the left ventricle. So in fetal circulation, it doesn't really matter what is right heart and what is left heart, because oxygenation really doesn't matter with the lungs, because it's not where we're getting our oxygen. So there's this structure, there's a gap between the right atrium and the left atrium. It's called the foramen ovale. And at this point, it is patent, it is open. At birth, when we start having oxygen in our lungs and we start breathing our own, right and left heart matter. And so the foramen ovale will close off. And so in our hearts, that hole is open. And so typically, it does close, but on occasion, the foramen ovale struggles to close. And that when it struggles to close, it's called a PFO, or a patent foramen ovale. And a lot of times you'll have the mixing of oxygen-rich, oxygen-poor blood. And a lot of times in babies, that is very dangerous. The baby could become cyanotic, and it can have a hard time, with the arterial blood and throughout the body, which should be delivering oxygen to the tissues, isn't doing an adequate job because at the level of the heart, you're accidentally sending out oxygen poor blood to the rest of the body. And so typically you can live with this. A lot of times if you do open heart surgeries on babies, because of a quote, people say hole in the heart, that's what they're talking about. But I've actually had a patient quite recently who was feeling short of breath. She had asthma and whatnot. So, people thought it was her asthma. But we treated her asthma and it wasn't getting better. So I had a cardiac workup. And so went to the cardiologist. Cardiologist agitated some saline in the vena cava, so in the right atrium. It's called a bubble study. And so as they agitated the saline, they watched the agitated saline move. Typically, you just go from right atrium to right ventricle, right? But they were seeing that the agitated saline was present in the left atrium. So we figured out this woman was in her late 70s and feeling short of breath, that, hey, she actually has a patent foramen ovale. So they went in, repaired this PFO, and shortness of breath disappeared. It was as simple as that. So you can actually live in the late stages of life with a PFO, but a lot of times you have to do surgery in a baby. Yeah, she was like 78 or something like that. So he was up there. So let's see. There's a question I think I missed. Does this mean that we have more than one shriveled up ductus venosus? You have more than one baby? No, because this is going to be attached to the placenta, and the placenta leaves the body at the end of pregnancy. So you only have one ductus venosus, and that is where your umbilical cord was attached. Otherwise, it's attached to the placenta. You don't need to know the pathway of fetal circulation / blood oxygenation. What you need to know is that there are three structures: ductus venosus, foramen ovale, and ductus arteriosus. The ductus arteriosus, let's talk about that, exists between the aorta and the pulmonary arteries. So you just need to know that these three structures exist, that after birth, they close up. The reason why they exist, we talk about the ductus venosus, is so the blood supply can get into our blood supply. But the PFO, the foramen ovale and the ductus arteriosus exist because right and left heart don't matter, because oxygenation doesn't matter at the level of the lungs, because that's not what we're getting. That's not what we're getting at. So these ducts and openings, they close. So that is what you need to know. The ductus arteriosus exists. I think I have another slide, kind of shows it better. The ductus arteriosus typically our aorta would have oxygen-rich blood, and our pulmonary arteries have oxygen-poor blood that send it out to the lungs. But because during fetal circulation, again, the lungs don't matter, right, left heart doesn't matter, there is a gap between the aorta and the pulmonary arteries. And that itself is called the ductus arteriosus, which can, it closes up, can on occasion remain open and whatnot. There's a movie, the guy who does Professor Snape, Alan Rickman, actually acts in it. It's in the early 2000s. It's about doing surgeries on babies that have patent foramen ovales, patent ductus arterioses, and whatnot. So if you ever want a study break but still learn, watch that movie. I forgot what it's called. Okay, stages of early development. So fertilization, right? The sperm meets the egg. So we have two specific reactions. The first is the acrosomal reaction. If we go back to the morphology of the sperm, what slide was that? It was, we have the structure at the beginning of the head of the sperm called the acrosome. So the acrosome is going to come into contact with the outer layer of the egg, and you'll create this acrosomal reaction. So the acrosome contains a lot of hydrolytic enzymes so that the sperm can essentially eat through the outer layer of the egg. Once the sperm comes into contact with the next layer of the egg, the sperm will enter into the cytoplasm, and you have a new reaction called the cortical reaction. So the cortical reaction prevents any further sperm from penetrating that same egg and causing way too many chromosomes. If you have two sperm that fertilize that same egg, the egg is no longer viable. Why is it no longer viable? Because the only viable chromosome count is 2n. If we add another sperm nucleus in there, you'll have 3n. 3n is not viable for humans. And so an egg that has two sperm that have fertilized it is not viable. It will not differentiate. It will not become a zygote. So this cortical reaction prevents any further sperm from fertilizing this egg. So it's a common misconception to think that twins occur because two sperm fertilize the same egg. That's not what happens. So there's fraternal twins, which exist because you have double ovulation. So each egg that gets released gets its own sperm that fertilizes it. So fraternal twins share a womb, but they are genetically unique. They are not identical because they have different eggs and different sperm. But let's say, how do identical twins occur? Identical twins occur after you do have this fertilization, you do have the cortical reaction. Only one sperm fertilizes this egg, but once you start doing all those cell divisions, you accidentally, one of those cell divisions, the cells don't stick together and they accidentally split into two. So it's the same exact sperm and same exact egg that have accidentally caused two separate dense ball of cells. So once you do have the cortical reaction and you have the sperm inside the egg, that's when the egg comes out of metaphase 2, completes meiosis 2, and that ovum will have its nucleus fuse with the sperm. And that's when you start to get the zygote and you get all that cell division. Ready. So cleavage, right? So once you do have that zygote, which is when the nuclei fuse, you start to get all these mitotic divisions without cell growth. So mitotic divisions, we are replicating the genome completely. We still have 46. Meiosis basically halves our chromosomal count. Mitosis just replicates all 46 chromosomes. So we'll have, during cleavage, mitotic division. So all of these cells will have 46 chromosomes, but the cells won't grow, right? This ball contains the same volume as this ball, contains the same volume as this ball, contains the same volume as this ball. But we still have more and more cells because you're replicating all the genome, but the cells themselves in between replications aren't growing. If you only have one egg is released during the cycle, how do fraternal twins arise? Is that from two eggs are being released? Yeah, so during double ovulation, an ovary might accidentally send off two eggs rather than one. So that is typically, quite frequently, twins run in the family. So if you ever realize that if you have twins, there's a genetic thing where different generations have twins, and the reason why is because there's some genetic inherited trait where sometimes the mom will double ovulate and will send off two eggs rather than one, and that's how fraternal twins arise. It's during that same cycle. So if you kind of go back and look at double ovulation, where is this? It's up here more. right? During ovulation, when one follicle is released, double ovulation is during this happen, 2 follicles are released. And so 2 eggs get sent off, and they both can get fertilized, and that leads to fraternal twins. Alrighty. So blastocyst formation. So as we keep having those mitotic divisions without cell growth, we create that dense ball of cells. The dense ball of cells is called the morula, and the morula hollows itself out and creates the blastocyst. And the blastocyst itself is what implants. So once we do have that hollow ball of cells called the blastocyst, and it has implanted, you do have folding, and these cells start to migrate, and these create different germ layers. So these different germ layers, you create 3 primary germ layers. The innermost layer is the endoderm, the middle layer is the mesoderm, and the outer layer is the ectoderm. After gastrulation, we'll talk more about the different germ layers and what arises next slide. But after gastrulation, we have neurulation. So the layer of the ectoderm will fold in on itself and create the neural groove and thus the neural tube, and it will give a rise to the brain and spinal cord. So this is a layer of the ectoderm. And a certain area of the ectoderm will be labeled the neural plate. The neural plate folds on itself, creates a neural groove, creates the neural tube. The neural tube itself is these stem cells will give rise to the brain and the spinal cord. But in terms of, for the most part, endodermal stem cells will only create endodermal tissues. Mesodermal stem cells will only create mesodermal tissues. And ectodermal stem cells will only create ectodermal tissues. The only to that rule are these green cells right here, neural crest cells. So they are technically an ectodermal stem cell, but they can essentially reprogram themselves and create mesodermal tissues if needed. So what are these tissues? So the major structures that come from the germ layers, the endoderm, which is the innermost layer, will create the guts, the lungs, and the digestive internal organs. Will create the inner tube of the tube within a tube system. The mesoderm, the middle layer, creates the blood, muscles, bone tissues, including the kidneys and the gonads. And then the ectoderm is the outermost layer, creates the skin and the nervous tissues, right? We have that neural plate that starts to form the brain and the spinal cord. So if you want to kind of break that apart, these different layers, you don't need to know this middle line right here, dorsal, paraaxial. I don't want you to know that. This diagram is here to show you that different organs, different types of tissues arise from different germ layers. And then you also have, some people consider this a germ layer as well of the gastrula, the germline stem cells, right? The sperm and the egg. Some people include this in it, some people don't. Mainly it's just the ectoderm, mesoderm, and endoderm that you'll see. But that is it for Repro and Dev. I finished 15 minutes early, so we can go over the questions and still get on time. If you don't want to stay for the questions, that is perfectly fine. They're not passage-based. These are more standalone ones, just to make sure you guys got a lot of the standalone, just gotcha type information that I've seen quite frequently on Repro and Dev. So if you don't have any questions or don't want to stay, you're free to go. Have a great rest of your weekend, and next week will be a little bit tough. That is the toughest of the physio lectures. But if you do want to stay, you don't. So do we need to know stuff about the chorion and stuff about reproduction development chapter? So you don't need to know about the chorion, the amnion, different layers of the placenta. That is just detail which not high yield, not something that I would waste time trying to memorize. Again, what you need to know is in the first few slides. I'm trying to kind of keep what's relevant on these slides. Use this as a study guide. So do you need to know about totipotent, multipotent, and pluripotent classifications? No, off the top of your head, they would tell you. I've seen maybe one or two passages on it, but they always tell you, what the difference is between the different type of stem cell classifications. Pluripotent, you can differentiate into a bunch of different things. Multipotent is you can differentiate into couple different things, but within the same similar type of tissue. That's not something I recommend knowing. Stem cell research is not really on the MCAT that much. Stem cell research is very interesting. And if you guys want to stay, we can go into different niches and talk about stem cell niches. That's always fun. But that's not something I want you guys to worry about. Let's see. Is the spinal cord made from ectoderm or mesoderm? Is it the dorsal notochord? Yeah, so the spinal cord is made of ectoderm. So it is the ectoderm that folds in on itself. It's just that these neural crest cells can bounce between ectoderm and mesoderm. And so what the spinal cord actually comes from, or the dorsal, which leads the dorsal to the dorsal notochord, is this neural tube, which is an ectodermal tissue. It's just that these stem cells, neural crest stem cells, can bounce between mesodermal and ectodermal, and these guys can lead to spinal tissue. It can lead to maybe some muscle, some vertebral tissue, if needed. It could bounce around. So that's something that's it's a unique type of stem cell that exists that is undergoing quite a bit of research right now as we try to figure out how to reprogram our own cells and create regeneration. So I could talk about that as well, but I'm trying to keep it relevant, All right. Let's get through the questions so we can actually finish on time today. In which of the following locations does spermatogenesis take place? Answer in the chat. I want everybody to get all these answers. Answer in the chat when you get an answer. Alrighty. What this is basically saying, where does sperm production take place? Seminiferous tubules, right? Afterwards, it gets stored in the epididymis, leads out through the vas deferens, goes through the ejaculatory duct, then nothing for if you're using 7-Up, and then out the urethra, and then the head of the penis. So 7-Up, right? It starts, gets produced in the seminiferous tubules. Next question, what is the purpose of adding alkaline fluid to the semen? Lovely. So just right off the bat, what's the purpose of making semen more basic? And that is because the female reproductive tract is acidic. And so while D does not say explicitly that the female reproductive tract is acidic, adding that alkaline fluid would help in their survival because it essentially neutralizes that environment. A, sperm maturation develops in an acidic environment and therefore requires basic fluid to counteract it. Evolutionarily, that doesn't make sense. Use your mental toolbox. Why would we mature something in an acidic environment and then give it a basic fluid? If that was the case, it would just pretty much, if it needs that basic fluid, you just kill it in the acidic environment in which it's maturing. That really just is counterintuitive. B, seminiferous tubules are slightly acidic and needs basic fluid to buffer sperm survival. Pretty much saying the same thing as A. And then C, acidic mucosal fluid is added to semen from other glands, not basic fluid. So personally, when I took the MCAT, answers like C always tempted me. I always want to be like, they're trying to trick me. So C is always one of those questions that, well, answers that I get hung up on because I just want to, not get caught. But it is D. It doesn't overtly say that the female reproductive tract is acidic, but that is what it is implying. Which of the following hormones released from the anterior pituitary stimulate the ovaries to produce estrogen and progesterone? Alrighty. So this is where we have to keep hormones separated based off of hypothalamic and pituitary, right? The hypothalamic hormone is GnRH, but what this question is asking is released from the anterior pituitary. So if it's released from the anterior pituitary, it will be the FSH and the LH. There's a question that was a little bit off topic, but we can answer it real quick before we go on to the next question. What is the basement membrane? You always hear that and get confused. So in skin, the basement membrane is essentially where the skin stem cells are attached. And so the basement membrane is where your epidermis, which is a lot of dead skin, is attached. And so it's just one of the layers at which the dermis and the epidermis are attached to. The layer of skin. And the last one, primary oocytes are arrested in the first meiotic division until what process occurs during puberty. So this one usually gets people. And we'll go over. All right, so let's go through that. So primary oocytes are arrested in the first meiotic division until what process occurs during puberty? So this is one of those questions that the wording, adding in jargon, makes you be like, it kind of just throws you for a loop. It's when you see primary oocytes and you see arrest, you think, oh, what thing sends things out of arrest? And in this case, you're just like, oh, primary oocytes mature into secondary oocytes during the follicular phase with FSH and whatnot. And so it kind of starts making you second guess, like, oh, are we talking about FSH? Are we talking about LH? What's going on? And so essentially what this question is all primary oocytes are arrested until we start kicking off the menstrual cycle during puberty. What kicks off the menstrual cycle during puberty? The LH surge, right? So all of the primary oocytes are going to be arrested in the first meiotic division. They're all going to be stuck in prophase 1 and their follicles until puberty occurs. And what's kicking off that puberty? LH surge. So if we talk about A, what's A? Oocyte fertilization. Oocyte fertilization is where it comes out of arrest of metaphase 2. It's in metaphase 2 at that point. It's already the secondary oocyte. So the key here is also first meiotic division. Oocyte fertilization, we're already in the secondary, the meiosis 2. We're at metaphase 2. We're just halted there until fertilization occurs. Oocyte implantation. Oocytes don't implant, right? Blastocysts do. Then oocyte maturation. So this process of leaving arrest is oocyte maturation. But that process is after primary oocytes leave arrest. So this can't happen before. So usually people choose between A, C, and D. Oocytes you leave because they're at rest metaphase 2, but they're already outside of the first meiotic division. So it is LH surge. This question is basically asking what kicks off puberty in a sense. And that is what sucks about the MCAT, right? That question could have been much easier. What kicks off puberty? But this is essentially that's all this is asking. And that what kicks it off is the LH surge. LH surge also causes ovulation. So keep that in mind. And that is it for today. So hopefully you guys enjoyed Repro Dev, got you out of here on time. It is a beautiful Saturday. Enjoy the rest of your weekend, and I will see you next week for endocrinology and immunology, which is the hardest, most dense of the physio. So this was a nice little intro before we head into that. Yes, we could do passages in future classes. I usually do passages in the old ones. For this one, there's not a ton of passage-based things. I'll add in some hormonal passages next week, if that'll help, because for Repro Dev, it's the hormonal control. That's the biggest thing. So I'll add more hormonal type things into next week's lecture. Awesome. And then there was a question. I don't know if you're still here. Let me check. There's a question on OTC ovulation kits. And honestly, I have never looked into over-the-counter ovulation kits. It might be testing LH levels. Let me actually look at this up. Let's see. Yeah, it detects LH surges. At least this one does. This one gets 99% accurate on Amazon. It detects LH surges because LH surges indicate you have reached ovulation. So that is answer to your question. Detects the LH surge. Any other questions?